the future of cancer immunotherapy headed anyway? What are the key ongoing trials, for example? Are there novel mechanisms out there? Are there pathways that most docs don't know about that are being investigated?
I think this is the most exciting area in cancer research right now. As Jeff alluded to, there's a lot of targets we haven't hit yet. We've just started hitting some of these checkpoint inhibitors. We've just started, this is our first cellular immunotherapy that the FDA has approved. I think we're really in the early years now of immunotherapy, but what's critical is the credibility that has been established by these therapies in terms of overall survival benefits and long-term disease-free survival benefits with the checkpoint inhibitors. How we build on those findings is going to be critical. Precision is going to be important. Recognizing for individual patients what those checkpoints might be or what those antigens might be. Recognizing newer clinical settings. Right now, where are we developing these drugs are in the most metastatic refractory settings, but where we envision immunotherapy working best is probably in the more curative settings of adjuvant settings or minimal disease state settings. So I think there's both a, I hope, a change in the population of patients we can treat, as well as the armamentarium we have to treat them with in a precise way, directed way, that will make it rational, effective, and hopefully real value.
Well, we're here, this is Chicago, and we're here at ASCO 2013, so everybody's here. The cab driver I spoke to coming in from the airport said, what are all these docs doing here? What they're doing here is exactly this, right? They're looking at cancer therapy. They're all excited. What are some of the key immunotherapy trials that are being presented here right now?
Well, you're going to hear a lot of excitement tomorrow at the Clinical Science Symposium about PD-1. There'll be 4 presentations. I'm lucky enough to be giving one of them. There'll be 3 on different PD-1 antibodies and 1 on a PD-L1 antibody, which also blocks that pathway. But you'll also hear follow-up on a number of ipilimumab trials. You'll probably hear a little bit about adoptive cell therapy for melanoma. You'll hear about new combinations of ipilimumab with GM-CSF, suggesting that perhaps you can boost the activity of IPI. There'll be some posters from my group talking about other combinations. You'll hear a poster or see a poster where we actually are testing PD-1 antibody as an adjuvant. So Dan's right, the ultimate test of whether an immunotherapy really works is, does it prevent recurrence after you resect a patient and render them free of disease? Everyone agrees that's the perfect scenario in which to test a vaccine or an immunologic treatment. So those are some of the things you'll see. And you'll see a host of abstracts on PD-1 antibodies in lung cancer, renal cell cancer, other histologies, suggesting that immunotherapy is not just for melanoma. It's now got respect. It's active in other histologies.
What was the crossover effect of the IMPACT trial?
So the IMPACT trial was the pivotal trial that led to the FDA approval for Provenge in advanced metastatic prostate cancer. And patients on that study, it was a unique design where originally the primary endpoint was progression-free survival. And then it was later amended to be a co-primary endpoint with overall survival. So the original study design allowed for a crossover. 2/3 of the patients were randomized to Sipuleucel-T, the investigational agent, Provenge. 1/3 of the patients were randomized to control, which was the leukapheresis product that was unmanipulated. And a portion of that was given back to the patients as a control arm to maintain blind. Patients who then met the criteria for disease progression were unblinded in that arm and given the option for a crossover study where those original leukapheresis products were then made into the immunotherapy, the Provenge-like frozen product, and given to patients. 2/3 of patients crossed over to that treatment arm, and those patients ended up having a longer survival than the control patients who didn't cross over. Now, there's a huge selection bias in that a lot of those patients who didn't cross over very well may have had more rapidly progressive disease that then required chemotherapy or other palliative agent therapies instead of that crossover product. So we have to be careful about interpreting it. But importantly, that group didn't do worse. There wasn't a worse outcome to getting that frozen product in either delaying chemotherapy or any other treatment effect. And if anything, it looked like those patients still had a very good outcome. It's possible that if there was a therapeutic benefit in some way to that product, that it could have, to some extent, decreased the overall survival benefit seen with this study. Now, it still met its primary endpoint. It still showed a statistically significant overall survival benefit despite that crossover. But if that had therapeutic effect and it wasn't there, we may have seen even a greater benefit.
And why don't you elaborate a bit on the Psa Quartile Trial?
Yeah, so a lot has been done with this study to try to understand exactly who is benefiting the most from Provenge. Mike mentioned it earlier. He said, I want to know who's benefiting from this therapy, because that's really who we want to make sure gets the therapy. Well, there's been a lot of sort of what we call post-hoc analyses, and one of them was to look back at the baseline characteristics of the patients that went on to Provenge in that study, and to see if there was any difference in outcome based upon those baseline characteristics. And one way of looking at that is at PSA. PSA is a prognostic factor in prostate cancer. Patients with higher PSAs with metastatic castrate-resistant disease do worse than patients who have a lower PSA. The lower PSA patients tend to live longer. So, and indeed in this study, when you break it out by quartiles, the patients that were in the lowest quartile had the best overall survival. But interestingly, there was a survival advantage for Provenge over control in each one of those quartiles. But the greatest difference was seen in the lowest quartile of patients, patients who had PSAs of 22 or less. In that group of patients, they had a hazard ratio of 0.55. They had about a 13-month improvement in overall survival. Now, this is a subset analysis. This is post hoc. We can't, this is not in the label. To me, this is hypothesis generated, but it's consistent with our thinking of immunotherapy. And that is that the patients that are going to benefit most are the patients that have the best overall expected survival, the patients that are going to live 3 years or longer, and the patients that have a lower overall tumor burden, which you would surmise would be in that group in general. So Peter, I think it's very reaffirming to me that the data is consistent with the rationale on how an immunotherapy such as Provenge should work.
It's fascinating.
Yeah.
Thanks.